the last speaker, we have Ron Wehrens. Ron got his PhD in chemometRics, and he's been professor in Twente and Nijmegen, I hope to pronounce it correctly, in the Netherlands. He also worked as the leader of a biostatistics and data analysis group in a research foundation in Italy. And let me say that is a prestigious one also. I might act as an expert here because it's not too far from my hometown. And he currently is the business unit manager at Biometrics, which is a large group at Wageningen University. This group is gathering different quantitative scientists, broadly speaking, working on development of mathematical tools for biological processes. So really, Ron is not working on a food-related application, I would say, or with spectral data, but on a way broader range of topics such as omics science, ecology, genetics, and so on. His research has focused on producing knowledge from the data in the natural science framework, but with a specific attention to reproducibility, code sharing, computational and data analytics details. And so among his contributions, it's worth noting that he's the author of the PLS package in our software. And I would say that is something that a lot of us ended up using at a certain point in our life. And he's author and maintainer of other really useful R packages, also providing visualization tools, and is the author of the book chemometRics with R. So a lot of work in the area. And today he's going to give a talk entitled Reproducible Research, the R in chemometRics. So please, Ron, you have again a total of 45 minutes, and you can handle the question as you prefer. Thank you.
Excellent. So thank you, Alessandro. Can everyone see the slides?
Yep.
Can you hear me okay? Yes, probably yes. Otherwise, you can answer the questions. That's a double one. So let me start by thanking the organizers for inviting me to present a part of my work. At least initially, I felt a little bit apprehensive about accepting, because I was basically saying, well, there's not too much work in scientific fields that I'm actually doing at the moment. But then Alessandro kindly invited me to talk about the reproducibility thing. And I must say, I was very, very happy to see so many of the topics of today. So there were a couple of things that basically reinforce the message that I'm trying to give in a way. And maybe before I start my own lecture, let me give you a couple of the things that I found extremely interesting in the presentations that I saw today. So first of all, Tom gave a beautiful overview of the history of chemometRics. But the most salient thing I thought was that he basically said lots of things work. And that's exactly the experience that I have too. And that's also what we saw in some of the presentations later today, especially the one by Valentina and just now. So basically, to give a little bit of background in my current work, I'm basically leading a group that does contract research. So our main aim is always to give an answer to the question of our client. And in many cases, we are happy if an answer can be given that is good enough with a minimal amount of effort. And what is also important is that we should be able to explain the reasoning behind our answer to our clients. So we tend to stay away from cutting edge research because it would mean a lot of additional explaining, unless that cutting edge research is really needed to produce an answer of the desired quality. Having said that, the second thing that I found very intriguing in Tom's presentation was his mentioning of the driverless car in the final slide. And that is something that I think is also a downside of the open source community. So there is this tendency of simply downloading every single package that is on CRAN and then trying out everything. So before you know it, people are generating results with tools they don't really understand. And that's something that's kind of scary. And I think that's the bad way of having driverless cars. So what we always try to do is to have statisticians who have a feeling for the subject at hand, talk to the experimental scientists, and then together work out a solution that works best for the data at hand and the problem at hand. And then Scott gave an interesting presentation about deep learning. It's something we have been using in the area of image processing, where it really has changed the playing ground. And to see a non-imaging problem recast as an imaging problem could be an interesting strategy to benefit from the strengths of deep learning. So I'm very interested to see what comes out of that. Claire had a really interesting point where you would really like to combine spectra and additional covariates. And that's something we have been encountering time and time again. And we haven't really been finding a very satisfying solution in the area of supervised analysis. So maybe the Bayesian PLS could be a really good way to tackle that. What we have been doing is we have been working with self-organizing maps, creating basically different distance matrices for the different data layers, if you want. So the spectra could be one data layer, and maybe another group of variables could be another data layer. And then if you take weighted sums of the distances in the separate data layers, then you do get quite interesting visualization possibilities that are also implemented in self-organizing map software that is on the CRAN. And then basically Valentina gave some interesting work on genomic selection that was completely in line with the results that we have been having in genomic selection and genomic prediction for plants. And also there we've been playing around with several different methods. I think mixed models are standard tools that we use, but we have been looking at rich regression and the SU and PLS regression as well. And the differences were not all that great. So in practical terms, I think we're back to the quote from Tom, lots of things work, and then that's definitely the case here too. Okay, but now coming to the topic of my lecture, reproducibility, that's going to be more and more important. Many journals want you to submit not only data, but also analysis scripts in order to convince referees and readers that what you have been doing is okay, or at least to give them the opportunity to reproduce what you did. And so let's see what practical consequences that has. So maybe I think the motivating example for me was this, and I'm sure many of you will have had the same experience, that you embark on a project, you do some analyses, you get quite good results, you write it up in a paper, and then the referee says, well, that's great. Could you maybe redo the figure in a different way or add something? And then suddenly you're no longer able to exactly reproduce the numbers that were in the original draft. It's usually not a big thing. So it's usually, I mean, digits at decimal places that are not really important, but still it's something that kind of is unsettling. And before you know it, you start wondering, well, maybe it's a coincidence that I had such good results, and you would like to at least be able to get exactly the same things back. We are already in a reasonable position because I think most of us will use tools like MATLAB or R or Python, so basically script-based analysis methods, which at least allows you to rerun the same scripts. But if you would be using point-and-click software, then it would be even more difficult to retrace your steps and obtain exactly the same results. But at least in the way that I used to work, I had separate directories for the data, separate directories for the scripts, and then I was collecting the results somewhere, and then the paper was in yet another directory, and then I was moving things around and making symbolic links and things like that. And when you start collaborating with people and you have a shared disk, then this problem will become only worse. And then there is the additional thing of giving appropriate names to your files, right? So I'm sure everyone has done things like first version, second version, final version, final, etc. These things are all part of setting up a workflow that allows you to retrace your steps, do reproducible work, and basically do high quality research. Well, the labeling point, so giving appropriate names to different versions, is basically completely made obsolete by using some sort of version control. SVN used to be the standard. Nowadays, I think most people will use Git. And if you don't know what Git is, then find out, because it will change your life in a very good way. It's quite easy, but I'm sure most of you will already know it. I think I saw a reference to a GitHub R package in the talk by Claire already earlier today. So that means Git is more or less a well-known package, or a well-known way of organizing data, scripts, and also results. So I'm not going to talk about Git, but I am going to talk about other ways of being reproducible in your research. And I chose PLS as an example. We've seen more presentations than I anticipated about PLS, but of course PLS is the chicken with the golden eggs of the area of chemometRics. Okay, I don't have to tell you what PLS is. So basically, you relate a multivariate X to Y, where Y may also be multivariate. And the key question very often is that you have to decide on the number of latent variables in your PLS model. And that is usually done by cross-validation. Again, it's mentioned so often today that I feel I can skip this slide without any further explanation. And in the R implementation from the PLS package, that is simply done as, okay, we have a data set where we want to predict the density of some yarns from their NIR spectra. And we are willing to use PLS components with up to 6 latent variables. And we are going to use cross-validation with 5 segments to decide on which of these different models would be the optimal one. Now, the PLS-R function also returns information about what the quality of the model would be if you would have no components whatsoever. In this case, that would be an RMSEP of 27. So it does say RMSEP here, but it doesn't mean that's the root mean square error of prediction. It's simply the function name. And here you say that it's about the cross-validation estimate that is part of the yarn.pls object. But what you see is that the first component already decreases the error, the second decreases it further, and the third decreases it even further. Fine. But what happens if you repeat the analysis? You simply do the same command again, then you get different results. And the reason is that the division into segments is a random process. So the objects, so the records in your data set that were in the first segment in the very first model are not the same records as the ones that are in the first segment in the second call to the PLSR function. And you can see that the intercept doesn't change, which is logical because it's a constant, but you can see that the other numbers do change. So the very first latent variable gives an RMSE of 8, and in the second model, where we have exactly the same call, we have an error that is slightly bigger than half of that. So that's quite a huge difference. And if you do that 100 times, you get this plot where you see the overall average, so the zero component solution as the blue circles, which is obviously constant, but you see that the variability of, in particular, the first component is quite large. Right, so that means that you have to be aware of the fact that if you do just one cross-validation, the actual numbers will change sometimes quite dramatically, and that also will influence your choice of the optimal number of latent variables. Well, you say, well, maybe we should use a statistical test then to see whether a model with 2 latent variables is significantly better than a model with 1 latent variable. And yes, such a test exists. there is a permutation test in the PLS package present in the function select ncomp. And if I apply the permutation test to the first model that we fitted, it says that 4 latent variables is optimal. But if we apply it to the second model, it says 5 latent variables is optimal. So again, here the statistical test is subject to some variability again, because also the permutation test is, of course, a process that depends on random inputs. Now, I have to admit, I had to juggle around with random seeds in order to get this difference. So in most cases of applying this function, we would have 4 as the optimal number of latent variables. But the point I'm trying to make is that you should not trust the outcome of such a function as something that cannot change. And you should be aware of the points in which randomness is injected in your analysis. And sometimes the results may differ quite a bit. And also, if you look at these curves, then I'm sure if you would ask 4 or 5 people how many components they would choose, their answer would ideally not only depend on what you would like to predict, but also what the desired level of error is. If you could live with an RMSEP of 10 or 15, then you might be inclined to even go for 1 latent variable in order to be as limited as possible in the number of degrees of freedom that you are using. So we know that data analysis is perceived to be a thing that should be completely reproducible, but there is variability, and I think it's not dramatic in the sense that that there is a large number of papers that should be retracted because if you would redo it, the results wouldn't hold up. I don't think that's the case, but we should look at this with kind of a common sense, and that's also the way that I always tell students how to look at the estimate of the number of latent variables that you should use. I thought it was remarkable that most of the speakers today didn't put too much emphasis on this choice of the number of latent variables. And I think that's in a sense okay, but there is a danger of automating this choice. So again, the driverless car here is maybe a bit too far, and I would always really look at the context of experiments and see what is needed and what the data look like. Of course, if you do large-scale comparisons where you have tens of different traits or data sets comparing to tens of other methods, then you have to have some kind of an automatic rule to choose. But you have to keep in mind that this choice actually influences the results quite a bit, as we also saw in Valentina's presentation, where she showed that the PLS models with very high numbers of latent variables were not doing so well. Okay, so what can you do, maybe not to avoid this lack of reproducibility, but to keep it in check? Well, the first thing I already mentioned, if you're using a script-based analysis tool, that is already good, because it allows you to redo your scripts. If you use R packages, either written by yourself or by others, that helps, because it basically unifies collections of scripts, puts in structure. I also think that the package system of CRAN is one of the main reasons why R has become so big. It at least enforces a minimum of documentation and a consistency within the packages. So that's not to say that every package is basically implementing something that is useful, but at least there are some errors that are being avoided. But also for yourself, if you have a collection of scripts, that's much harder to maintain than if you have an R package that you can test using the standard tools that are provided by R itself. One further way to improve things is to integrate your reporting and your calculation. And the first system that allowed you to do so was the S-Weave system by Friedrich Leich from 2002, which integrated R and LaTeX. So basically what you did is you made a LaTeX document that also contained R code. And in the first compilation step, the R code was translated into LaTeX code. And the second compilation step translated the LaTeX code into something like PostScript or PDF. This was further developed in a package called Knitter by Wehrens, which integrates R and LaTeX, but also Markdown, and it not only can be used to make PDF documents, but also it can be used to make HTML output so that you can show the results on a web page. And again, you can go one step further and use Jupyter notebooks, which are completely web-based, and they are not just there for R, but you can also use them for Python, etc., etc. I don't have too much experience with the Jupyter notebooks. I got stuck at the Knitter level, so that's what I'm going to talk about mostly. You can even go one step further and talk about Dockers, which are basically containerized pieces of software. So basically a snapshot of the system that you have in your computer that will be frozen and that will be shipped to the customer, so or the partners in your research project. And that means that everyone is using the same software with the same versions, etc., etc. And that should also lead to the same results every time that a particular piece of software is run. Again, not something I'm going to discuss today because that's more in the realm of computer science, but it is something that we use to ship, for instance, products that we make for customers. Okay, so to come back to Knitter, again, this is code as well as text. So R code in this case, the code execution leads to results that can be R outputs, it can be tables, it can be figures, and the integration lead to a document like a PDF. Actually, the presentation that I'm showing you today has been made with Knitter in exactly the way that I'm trying to explain to you now. And for instance, if you need the R code that has been used in the paper, then you can easily extract it with one R command, and you basically get an R script containing everything that is needed to reproduce the results in the paper or in the document. Yeah, one very useful thing is there is the command session info in R, and that is something that I usually include as a very last page in a report, because it tells you everything you need to know about the R versions that are the R version that is used, the packages that have been used in the analysis, etc., etc., and also the operating system information and the platform on which you run it. I don't think clients or partners will usually read this unless there is a real need to explain differences, and then it's very handy to have this information available. Okay, so I would like to give you a short case study. Unfortunately, I haven't been doing much spectroscopic work in the last couple of years. I was mostly involved in Metabolomics type of projects, but this was the closest I could get. Actually, I'm currently working on 2 spectroscopic projects, but they are not far enough to give you any results. But basically, I think that this project will give you the flavor of what I'm trying to say, and give you also the an impression of what kind of things you can expect from such an approach. So what we are trying to do here is we are investigating alternative sources of proteins for humans. So as you probably know, most of the human protein intake is coming from animal products, and one way of making our society more sustainable is to eat more plants, basically. Problem is, most plant proteins are not very well digestible, and there are not too many plants that have a lot of protein in them. So a lot of research in Wageningen, at least, is looking at finding alternative protein sources for human consumption. Now, this is a small-scale project in which 12 subjects were subjected to a crossover design. They were given 3 different protein shakes, and their amino acid levels in blood were monitored for 3 hours. And then there was 1 week of washout, and then they would get the second shake, 1 week of washout, and they would get the third shake. And the idea was that we could compare 2 of the protein shakes were from vegetable material, and the other one was whey, which is something that is kind of a standard protein to compare with. And typically the results look like this. So here you see histidine, for instance, serine and leucine, and this is subject 1. You see after the admission of the intake of the shake, you see that after protein A in the blue line is going up quite strongly, whereas the green line is hardly increasing, and the pinkish line is showing a peak, but at a later stage than the blue line. And you can, if you look at the other participants in the columns, you see more or less the same behavior. And okay, so the idea was to use these data, these time points, to say something about the ease in which the protein could be digested by different people. Okay, so the first thing that we did is we wanted to go to an easier representation. So we had 8 different time points, I think, 2, 4, 6, or maybe even 10. What we did was we fit curves through the points, and actually these curves were so-called Wood curves. So is that in the next? No. So these curves basically were coming from dairy science, and they were used to describe lactation behavior of cows. They were developed by Wood in the 60s. And basically you have an exponential part, and there is an amplitude part, and D is the baseline. So basically these curves are determined by 4 parameters, A, C, D, and M, where M is basically the time to the maximum, C is related to the steepness in the beginning and the steepness after the maximum, D is the baseline, and as I already said, A is related to the amplitude. And once you have fit these curves, once you have these 4 parameters, you can actually extract parameters of interest, like the area under the curve, but also the time to the maximum that is already given by M. This is shown here. So for all of the 3 proteins, A, B, and C, we can see that here we see that the most AUC values are very small. So C is a protein where the AUC is rather small for most people, but it has a couple of large values, whereas the AUC of protein A, which is the reference protein, is much more widespread, and in general is larger than the other 2. Of course, we're interested in the direct comparison between the proteins, and for that we use mixed models. And so for each of these AUC values of individual amino acids, we can then generate an estimate of the ratio between the AUC of the protein and the reference protein. And what you see in most cases, for instance, for protein B, is that the estimate is smaller than the AUC value of the reference protein. And here we have the confidence intervals that go with it. And any confidence interval that doesn't include the reference line of 1 is indicated in red. So you see that for the vast majority of amino acids, the intake or the uptake of protein B is lower than the reference protein. Something similar is true for most amino acids in protein C, but there are also a couple that have seemingly larger values, although you can see that these confidence intervals are very large, and that usually has to do with very extreme values in the fit. We try to filter out some of the bad fits. You can see that in these examples, the curves follow the points reasonably well, but especially for the cases where the uptake is not very successful, sometimes we have bad fits. So we need to also do some kind of curation in order to filter out bad fits. Anyway, at least this approach gives us a very principled way of saying something about the efficiency with which different proteins can be digested by humans. In another project where we had a similar data set, we saw that some people were actually unable to digest proteins altogether, so they basically were not taking anything from that protein shake, whereas other people were maybe having a slower or a lower uptake, but at least they did get something. So there is also this responder non-responder issue in the population. In this particular data set, we didn't see any non-responders, so people who didn't take anything in. Okay, so the actual implementation, to come back to the topic of the reproducible research, is that we developed the methodology, so fitting the wood curves and doing the linear mixed models in a bunch of loose R scripts. The next step was to combine these in an R package AA response, and we also wrote a vignette. So that's the R terminology for a written tutorial that contains example data, and that is included with the package. And basically, in several new case studies, because these are case studies that are being continuously presented to our colleagues in the food department, we are using the functions from the R package, and we generate mid-R documents in which we explain our findings, but also in which we explain choices that we make about, for instance, excluding outliers or maybe repairing obvious errors in data sets. So for instance, once we had one particular time point that was measured really badly for one particular subject over all amino acids. Well, that's the sample that you really want to remove from the data set and replace by missing values, for instance. Once you start compiling such a document, you will have an R console open in which all the objects that you have created are accessible. So if you would have a question like the one of the referee, could you adapt your figure in a way that makes something else clear, you immediately have all the original data available that you have generated during the stepwise analysis of all your data. And that makes it very easy to make last-minute changes to your figures or to your tables or even to your conclusions. But also, if you find out that there are errors in your data, it's a very easy way to change it. For instance, one of the first data sets of this type that we analyzed, we saw that one particular subject, I think it was number 7, I'm quite sure it was number 7, had reversed colors. So basically all the subjects were having a much larger uptake of the reference protein and a much smaller intake of the test protein, but with subject 7 it was the other way around. So we immediately noticed that because we also do quite extensive visualization. And then the experimental scientists could go back to the lab and confirm that basically the files were switched. They were in the wrong position or something like that. And they could confirm that the actual values were the other way around. So the only thing that you need to do in such a case is to correct the data in the original CSV file and push the rerun button, and all the analyses and all the figures are being regenerated automatically. If you have outliers or you decide that some points are outliers, you can simply flag them as such, remove them from the data, and rerun. Again, all the analyses and all the figures are recreated on the spot. If you decide that a different type of curve would be more optimal than the wood curve, fine. Simply replace that function, change once, and rerun. And so there are a number of things that you can think of. The main point being that working in this way allows you to, at all time points, make the current state of affairs available to others as well. So we use this also in connection with the private Git repository. That means that several people who are working in the same data set can jump in at exactly the same point and have exactly the same information and maybe change things or maybe discuss things. So this collaborative research is also much helped by the NITR documents that allow you to really go from A to B in one prescribed way. Of course, what you need to watch out for is that if you change something in the data, then everything changes, but you should remember that the text doesn't change. So if you say it's clear that method A is working better than method B, you change something in the data and suddenly this is no longer true, you have to change the text as well. So usually we start by generating the figures and writing, and we write key parts of the text that describe decisions that we took, for instance, in data pre-processing or pre-treatment, that are related to the code, and the conclusions are only filled in after the analysis itself is deemed to be more or less constant. The second point is the point that I was referring to in my PLS example of choosing the optimal number of latent variables. So you have to be aware of where randomness is coming in. And if you don't set a random seed in your document, then the results will be slightly different every time. So I usually set a random seed, which allows me to exactly reproduce these numbers. But at the same time, that doesn't mean that these are the correct numbers in the sense that no others are possible. So we have, by setting explicitly a random seed, you also make visual, or make explicit, I think that's the word, that randomness is important at a particular stage in the analysis. Yeah, something that always bugged me when I was still not using NITR is that if you are not careful, you start an analysis with a workspace that is not clean. So you may have some debris from earlier analyses. So the very first command in my NITR document is usually clean everything, throw away everything that you might have. And then you really start from a fresh, clear environment and you really get the same results every time. Sometimes computations take time, and that can be a reason for saving intermediate results in binary files that, if present, can be used to avoid these computations. So you usually check if a particular file is present. If it is present, load it. If not, do the computations. This is also dangerous because you have to remember that each time that you change something prior to these computations, you should throw away the computations so that the calculations are redone. So again, there's no way of escaping the need for common sense and thinking. That your mind should always be switched on in these things. Unfortunately, maybe for some, but that's how it is. It does help in speeding up things. So if you're basically changing the layouts and you don't want to do the computations again, then this is a really good way of avoiding that. Okay, so basically the tools that I'm describing now have completely changed the way that I approach my research project. So 10 years ago, I was not using version control. I was using loose R scripts, data spread out in directories, and I consider myself to be quite an organized guy. But once in a while, I got into real trouble. So the very first thing that I basically started doing was version control, initially with SVN and later with Git. I started organizing my scripts in packages. That's also the reason why I am actually the author of several packages on CRAN, because once I have a package, I figure that possibly other people can also use it. and then I upload it to CRAN or to GitLab. Nowadays I use NITAR to bring together data, texts, figures, and basically what I'm trying to document is choices that are made, decisions that are made, striking observations that need explanation, and also some conclusions. And especially if you're working in a contract research organization, what I notice is that clients really appreciate such reports because they are also allowed to collaborate in the thinking process. They have something to discuss with you, and very often giving them such a report in a preliminary version leads to comments from their side that lead to a much better version next time around. So the fact that you actually make a document that serves as an endpoint of a stage in an investigation is something that I really like. And that's also the reason why I usually make PDFs and not web pages, because they are too fleeting. I mean, I would like to see the PDF as a physical, well, as a tangible object of the result of maybe 2 or 3 months work. And then if there is a follow-up, there will be a second report. Okay, that's the report for the customer. Actually, we have been discussing within the group that these reports also lead to a better quality, because once you write a report, you really often rethink what you have done. And at least to me, it happened several times that during writing a paper, I realized that I missed certain elementary things in the investigation and had to fill in the blanks. And that's exactly the same that can happen with writing a report for a customer or a partner, if you like. And exactly that particular report can be the nucleus of a paper. So it's not all wasted effort. Okay, I happen to write quite easily. Basically, I think while I write, I think better when I write than when I talk. So that may be recognizable for some of you. So my inclination is always to write down stuff because it helps me to focus and concentrate. But then when you do get to the point of turning that into a paper, it's also usually time well spent. Yes, and it's not just for small things. So Alessandro kindly mentioned my book. That's also done with NITAR. So this is a document of 300 plus pages. And so the second edition just came out last year, and I avoided using NITAR, making some of the errors from the first edition. So in the first edition, there were 2 or 3 figures that were not in agreement with the R code that was in the book. And that has now eliminated that problem. If you are more a person who likes to look at video clips on YouTube, I really could recommend you to look up Keith Baggerly. He's a guy who worked in the MD Anderson Center in Texas in cancer research. And he basically made a career out of what he called, I think, data analysis forensics. So he basically tried to reproduce the results from literature. And in many cases, he simply didn't succeed because the reports were not clear enough. And even if they added code, even MATLAB or R code, very often the results were not exactly reproducible. Again, most of the times the differences were not very important, but sometimes they were. And I think it's a very good, he has a couple of cases where he really drives home the point that this is something that is worth investing in and paying attention to. Okay, so I'm more or less at the end of my lecture. I would like to thank a couple of people, in particular Jasper Engel, who has been collaborating with me on these on the case study where we investigated the intake of protein shakes. Jos Hageman, Michael van Ouden, the other chemometricians in my group, Marco van Lente, who is our Git expert and who taught me all that I had to know, which is only 5 or 6 commands. That's fine. Diederik, Juriaan, and Aert are the other experts involved in the case study, as is the Wageningen University and Research Protein Investment Team. And I'd like to thank you for your attention. It's been a long day, interesting lectures, but I am sure I would rather be presenting at this point than listening. So I admire you for your stamina for still being with us. So thanks. I'll take any questions.
Thank you, Ron. We have some time for questions. There's a long comment now by Gustavo in the chat. I don't know if you want to read it by
Yeah, I will read it. That's probably
yeah, thanks.
Okay. Yes, so in general, if you want to impose quality measures, you will face some counter pressure. And in my experience, so people are not going to do it because you tell them to. They are only going to do it when they see the benefits and when they are convinced that it's also working for them. So that's one of the main reasons why I gave the talk today, because I hope to convince maybe some of you that this is worth investing and this is really worth doing. But you're absolutely right. I think the same is true for instance, sharing your data. I mean, if you would upload, so I mentioned I was working in the field of metabolomics for some time. There is a repository called Metabolomics. They have tools to make your metabolomics data sets into formats that can be fair, so findable, accessible, interoperable, and reusable. But these tools are quite difficult to use. And many of the metabolomics experimenters that I've worked with are very, very reluctant to spend maybe 2 or 3 days to make their data sets available to the community in that particular way. On the other hand, it's the only real way of doing it. So, and until the tools are improving and making it easier for us to do so, I think there is no other way as to go for it. And also management is partly responsible. So we face quite strong pressure in Wageningen University from the upper management to make our research reproducible, to make our data available, and in general to make our work accessible for the scientific community. And that absolutely helps because they also provide some resources to do that. Yeah, so let me see if I covered all. Which advice would you give to journals? Well, I think journals are also helping quite a lot. So there are quite a lot of prestigious journals that actually require you to put up your data in data repositories. And indeed, it would help if they would also require you to upload your data analysis scripts, which is something that I usually do. And I do see that referees appreciate it. So even if it's not obligatory, you can make your life easier and you get your papers accepted more quickly if you do it. So that's also a way to advocate this approach. So let me put the chat window next to my camera so that I appear to be looking in the camera.
Yeah, there's other 2 comments and question by Brandon and Georgiana.
Yeah, let me see. So Claudia responds, maybe that's I think the response to my talk or not. The rumor that predictive models are related to the expertise of the data analyst and the chosen method. Yeah, I think that I fully agree with that point. So I do see the value of comparing, let's say, new techniques with standards like PLS. But again, in applied work where you basically try to give an answer that is of sufficient quality within a given time frame, then you usually don't have time to do an exhaustive investigation, and there is usually also no need to. So in my experience, the limiting factors are usually not the algorithm that you use to give the final answer. I think very often I see that the data pre-processing is essential. So if you make a mistake there, there's no way that you can correct for that by choosing a better algorithm. The interaction with the problem owner, so let's say the domain expert, that's absolutely crucial, probably the most important thing. And then whatever model you use is, to my mind, at least in my everyday practice, of less importance. Okay. Gustavo, let me look at the comments.
I already answered
I
to that.
already did it, yes,
yeah.
thanks.
Yeah.
Brandon, many applications data come from databases and then the analyst only gets the data in some format. Do you have any experience where reproducibility is done all the way from the database to the final report? Yeah, this brings me to a pet peeve of mine, namely that nowadays with the surge of data science as a discipline, people seem to think that everything can be connected to everything, and that should always be good. This is not, in my experience, the case. So there are certain types of data that are very logical to be put in databases. For instance, protein sequences, DNA sequences, all that kind of stuff. Of course, there are errors in there, but that's something. I mean, the data still has some kind of general validity. The other extreme is, for instance, metabolomics. If you look at mass spectrometry-based metabolomics, the measurements from batch 1 may be measured on Monday are not necessarily comparable to the measurements from Tuesday, batch 2. So even there, you should really look into ways of making them comparable, and the best you can do is something like ad hoc approaches, which look at, for instance, reference signals that should be similar or equal in both batches. So data from databases is a moot point. I think if you can limit your comparisons to data coming from the same experiment, that's usually good. And then if you have several databases having several similar experiments and they all point in the same direction, then I think you have a point, and basically the data corroborate each other. I'm not quite enough of an expert to say something about, for instance, near spectroscopy. If these spectra are dependent on the device that they have been measured with, I expect they are to some extent, but I don't know whether you have any experience. I don't have enough experience to answer that question in that respect, but it's a danger, and you would have to check the assumptions that you have.
Thanks.